What's that way you possible people?
So
Other one bar.
I don't use them for my phone.
Yeah.
How many?
One bar.
How many percent?
It's a 28 now.
If it was up.
It was 17.
I don't do that.
All right. Okay, can you see my screen? Good morning. Can you see my screen? Yes. All right. All right. So we're just going to start because we don't have time. Please stop writing your names in the chat box. I will be able to pick it up from. Why is it echoing? Sorry, 1 second. No, why? Okay. Just down. Okay. Can you see my screen? If you can see my screen, please type 500. 500, good, good. Okay. I turned on my video today because I realized some of you were using math for me. I would normally turn on my video during my lectures, but this year I decided not to. So today, which is our concluding lecture, I've just decided to turn on my video and then just try to finish up. Right. So we're just going to dive in quickly because we are pressed for time. This is 8:05 already, and I have to finish up today. So we started looking at the structure of the mitochondrion in our last lecture, and I told you that there are 4 parts. Majorly, you have the outer membrane, inner mitochondrial membrane, intermembrane space, and the matrix of the mitochondria. And in the matrix, the mitochondria has its own DNA and ribosomes. So you would see ribosomes here, in the matrix of the mitochondria. So that way they're able to produce their own proteins, right? And because of their DNA, they also have the ability to replicate their own DNA. So you have what we call mitochondrial DNA. And when you think about inheritance, right? Mitochondrial DNA is something that we use, in forensics to resolve parental disputes, right? Particularly maternal, inheritance, because you inherit your mother's mitochondrial DNA. And so, this is just to tell you that the mitochondria has its own DNA, yeah? And then if you look at this, you also have the cristae. So from the inner mitochondrial membrane, there is some form of invagination that protrudes inwardly that is called the cristae, singular crista. And like I said earlier on the outer mitochondrial membrane, you can have porins, which are proteins, that are, integral membrane protein, but they are beta barrel sheets that are folded into a pore. And that's why they are called porins. Okay, so those are the 4 major parts of the mitochondria. And this table summarizes the functions, the metabolic functions within the mitochondria. So you have the outer membrane. Some of the functions is that it helps with phospholipid synthesis, fatty acid desaturation, fatty acid elongation, that is elongation of fatty acids. Fatty acid desaturation, is a process whereby enzymes that are called desaturases add double bonds into the saturated fatty acid chains. And then that converts them into unsaturated fatty acids. So you have saturated fatty acids, and then you have enzymes called desaturases that then add double bonds, to these saturated fatty acids to make them unsaturated, and of course that changes their physical properties, and it makes membranes more fluid because they are unsaturated. But in the inner mitochondrial membrane, you have things like electron transport chain, you have oxidative phosphorylation, pyruvate import, fatty acyl-CoA import, metabolite transport. But the electron transport is something that you really look at as a topic by the time you get to year 2, because it's also a way of generating more ATP. Protons are pumped into the intermembrane space and then eventually enters complex 5, which is ATP synthase. But for now, you don't need to know too much about it. So that's actually the major cellular respiration, but I thought that if we try to introduce you to that, by the time you get to year 2, when we'll be teaching it, it's just going to be repetitive. So for now, just note that it is in the inner membrane. Remember that I told you that the inner membrane is like a line. So you see, that line now is what we are saying, or is where this electron transport, takes place. And I've told you that that line is actually not a line, but it's a phospholipid bilayer. And so you have the matrix. I told you that it has its own DNA, and DNA can undergo replication because of the central dogma. There's what we call central dogma of molecular biology, where DNA undergoes replication, and then DNA undergoes transcription into mRNA. mRNA is then translated to proteins. Now, because the mitochondria has its own DNA, it can undergo transcription to RNA and then translation to proteins. Remember, we said that the ribosomes are also present and they're able to synthesize proteins. So, all of this happens within the matrix of the mitochondria. In the intermembrane space, what you basically have is the release of protons, pumped into the intermembrane space. So it's a proton, it has like a high proton gradient, and that's basically what the intermembrane space does. But the three other components, or compartments of the mitochondria are the outer membrane, and their functions have been specified here. And so let's move to the nucleus quickly. So, from high school, you also know that the nucleus is the largest organelle in the cell. It serves as the control center, because it stores the cell's DNA. It helps to regulate gene expression. So I just explained what happens in the central dogma in mitochondria, because the mitochondria has its own DNA, but your DNA is majorly found or stored in the nucleus. So if you look at bullet point 3, the major function of the nucleus is to regulate cellular activities, including metabolism, growth, division. So when we talk about cell division, we talk about mitosis, meiosis, all of that happens because DNA undergoes replication, and this DNA undergoes transcription. Talk about inheritance, everything has to do with the nucleus. So you talk about your chromosomes. I think Dr. Fakaridi already taught you, the number of the pairs we have. So we have 22 autosomes plus XY, or if it is a male, 22 pairs of autosomes plus XY if it's a male, or 22 pairs plus XX if it is a female. But these chromosomes are found in the nucleus, right? And chromosome number varies from organism to organism, species to species. So, and then you also have small spherical bodies called the nucleus, which are responsible for ribosomal RNA synthesis. So the nucleus is what produces rRNA. Please note, the nucleus in the nucleus is what produces rRNA, and then it's also responsible for ribosome assembly. And remember that you need ribosomes for protein synthesis, and these ribosomes are found on the endoplasmic reticulum. So again, the nucleus is very important. The nucleus, which is a part of the nucleus, is also important for rRNA synthesis and ribosome assembly. And then chromosomes, they are thread-like structures that are condensed, that carry genes. And these genes are the basic units of heredity. So when you talk about, of hereditary, is also correct. Basic unit of hereditary or heredity in organisms. So when you are asked what is a gene can be defined as the basic unit of heredity in an organism. And again, these genes are found, in the chromosomes, because chromosomes carry these genes, right? So if you're looking at chromosome structure, you have the long arm and the short arm. The short arm is called P, while the long arm is called Q. So P, sometimes you think of it like petite, P, and then Q. So it's just so, but all on these chromosomes you have genes arranged, some genes are on the long arm of the chromosome, some genes are on the short arm of the chromosome. So the nucleus, like I told you earlier on when we looked at the membranes, I said that the nucleus has two membranes. So it's a double membrane bound organelle. And by structure, it has a nuclear envelope. It is this nuclear envelope that has the outer and the inner membrane. So if you look at this figure, you see the nuclear envelope, which is here. This nuclear envelope is actually the outer membrane and the inner membrane. So you have the nuclear pore, you have nucleoplasm, like cytoplasm, and then you have chromatin, which is thread-like, that condenses into chromosomes. Then you have nucleus, which I said helps with rRNA synthesis and ribosome assembly. And then around the nucleus, you have is continuous. Remember when I told you about the endomembrane system, I said the nucleus is continuous with the endoplasmic reticulum. So if you look at this other figure of the nucleus, you would then be able to see the nuclear envelope, the outer membrane. So this is both the outer nuclear membrane and the inner nuclear membrane makes up the nuclear envelope, which is here, right? So this nuclear envelope is this, but then it comprises of the outer nuclear membrane and the inner nuclear membrane. And then you have the perinuclear space, which is the space between the outer nuclear membrane and the inner nuclear. Don't forget, there are different names. So the mitochondria, the outer mitochondrial membrane, inner mitochondrial membrane, intermembrane space. For the nucleus, you have the outer nuclear membrane, inner nuclear membrane, and then you have the perinuclear space. So they are different, right? So please note, then you have nuclear pores, and then nuclear lamina. In this nuclear lamina, you have lamines. They consist of intermediate filaments. So you have A-type lamines, B-type lamines, and other lamines. And they are involved, these lamines are involved in the functional organization of the nucleus. They are also important because they play a major role in the assembly and disassembly, before and after mitosis. So they are really involved in mitotic cell division. Right. So just note that in this nuclear lamina, you have lamines, A-type lamines and B-type lamines. So A-type lamines are also next to the nucleoplasm. So this is the nucleoplasm, while the B-type lamines, they are near the nuclear membrane, the inner nuclear membrane. That's where you would find the B-type lamines. So the functions of the nucleus, basically to store genetic material that are organized into chromosomes. So you know that DNA plus histones, the proteins, a protein called histone. DNA plus histone helps to organize the genetic material into chromosomes, and this ensures accurate DNA replication and cell division. The nucleus also helps to regulate the transport of regulatory factors. So you have things like transcription factors, mRNA, some of these proteins that I mentioned, even chromatin, all these regulatory factors, mRNA, and then gene products between the nucleus and cytoplasm via nuclear pulse. So during the central dogma, you have DNA being transcribed to mRNA, but this mRNA then has to be exported to the cytoplasm. It is via the nuclear pore that the mRNA gets to pass through, to go to the cytoplasm for translation into proteins. So, and that's what I have just said. So the nucleus is also important for the production of mRNA, which helps to, which is very important for gene expression studies. So if you want to know if a gene is expressed or not, what we are interested in is mRNA, because then the DNA has been transcribed to mRNA. And then you have ribosomal RNA that is being produced by the nucleus. So it's still a function of the nucleus. You know, there are different types of mRNA, of RNA, mRNA, rRNA, tRNA. You also have microRNAs, snRNAs, you know, so many. But again, you see that the nucleus is very important for the production of mRNA and rRNA. Then you have gene expression and duplication of genes, like I said. During cell division, this happens. So we're just going to quickly move to lysosomes. So again, in year 2, those of you who will do cell biology, these organelles are individual topics. So, but at this level, we just want you to please don't write your name in the chat box. Just, I will pull my attendance from the Zoom data, Zoom reports. All right, so the lysosome also has a Greek origin and is from 2 words, lyso and soma, or som, right? And lyso means lysis. So when you hear lysis, lyso, so to break down, and then soma, body. So lysosomes are organelles that help to break down, you know, different things. And like I told you when we're looking at the Golgi, Golgi produces lysosomes, or lysosomes are derived from Golgi, right? And they are also membrane-bound organelles. But lysosomes contain hydrolytic enzymes. Please note, they contain hydrolytic enzymes. Somebody asked a question the other time about peroxisomes. Peroxisomes contain oxidative enzymes. Please note, they are different. Lysosomes contain hydrolytic enzymes. Peroxisomes contain oxidative enzymes. And this, from the name hydrolytic, you can tell that they help to hydrolyze, you know, various molecules. So they function as the digestive system of the cell, also known as the stomach. So that's what the lysosome is. It's the primary site for the breakdown of macromolecules into monomers that can be reused. So they also help to digest the content of food vacuoles and vesicles, various vesicles. Lysosomes play a key role in autophagy, and autophagy is just a cellular process that helps to break, where the cell breaks down, you know, its damaged components, you know, unnecessary components, waste materials, things that it doesn't need, damaged organelles, old organelles. So when you talk about organelles that are old, you know, I told you earlier about senescence. So sometimes some of these things are old, and then it wants to break it down. There's what we call autophagy. Autophagy just helps to break down. And because of the hydrolytic enzymes in lysosomes, you know, which is due to a pH of around 5, which is very acidic, it helps this hydrolysis reaction to occur. And so the internal pH of lysosomes is 5, unlike the cytosolic pH, the environment of the cytosol, which is around 7.2. You know, so this helps the content of lysosomes to remain and to prevent cell digestion. So, because one might argue that why do lysosomes not lyse themselves? Because their pH of 5 is only within lysosomes, so they don't leak into the cytosol, right? And the pH of cytosol is 7.2. So because of that, lysosomes are able to stay in the cytosol, but because of their own pH that is acidic, and 7.2, of course, you know, that's around neutral, that's like a neutral pH from your pH scale, right? So because of this, they are able to remain in the cytoplasm or in the cytosol, and they don't get degraded by their own enzymes, lysosomal enzymes. I hope that you got that gist, because I mean it's very important. So the pH, the pH helps this organelle not to digest itself, but it can digest other old organelles. And I'm just going to show you this figure on the right that has the hydrolytic enzymes. So you have nucleases, proteases, glycosidases, lipases, phosphatases, sulfatases, phospholipases. So these are the enzymes that you would remember. I told you that the pH is around 5.0, but the pH of the cytosol is around 7.2. Of course, this is showing you the diameter of a lysosome is between 0.2 to 0.5 micrometer. And so nucleases, from the name nucleus, you can tell that they break down DNA and RNA. You know, DNAs. So nucleases are DNAs and RNAs, enzymes. Enzymes usually end with the suffix ASE. Amylases. Okay, so let's just start from nucleus. Nucleases help to break down DNA and RNA. Proteases help to break down proteins into amino acids. They digest proteins into. So, I mean, I used an enzyme. My internet is unstable. I used an enzyme called trypsin that helps to digest protein. So you do a tryptic digest experimentally from blood. So very recently, I worked on a sickle cell project and we had to extract proteins from blood spots. And then we used proteases to digest these proteins into smaller amino acids. So that's what proteases are. Glycosidases, they break down carbohydrates. Lipases, of course, lipids, right? Fats. And then phosphatases removes phosphate groups. Please note, phosphatases removes phosphate groups from molecules. Sulfatases breaks down sulfate containing compounds, you know, etc., etc. So if you look at enzymes, so these are the enzymes, and when there is a problem with these enzymes that I have mentioned, then what you have is that some of these unwanted organelles or undigested biomolecules, they will accumulate inside lysosomes. So if these enzymes are not active, if they are not present, there is going to be a problem. So the lysosomes will be filled with so many materials, and then it will swell, it will enlarge. And as a result, it will disrupt the normal cell and organ function. And this enlargement of lysosomes leads to what we call lysosomal storage diseases. Lysosomal storage disease, and these are severe and life-threatening. So, for example, an example of a lysosomal storage disease is known as Tay-Sachs disease, Tay-Sachs, and it's caused by the accumulation of lipids in brain cells. Imagine that your lysosome has so many lipids, you know, in it, and then it has enlarged, and so you have so much lipid in the brain, you know, and that leads to neurological damage, and it can affect children. When it affects children, they do not even survive beyond early childhood. So quickly, peroxisomes. I'm looking at time because we have a lot and that I want to discuss. Peroxisomes are enzymes that are filled with, they are small enzyme-filled sacs found in both plant and animal cells. So most of these organelles that we have looked at, you can find them both in plant and animal cells, you know, but the major ones I told you earlier on, you have things like chloroplast, cell wall present in plant, and I already told you to do like a comparison and similarities and differences between plant and animal cells, which of course I expect you to even know at this level. But when you, like I mentioned earlier on, the enzymes present in peroxisomes are called oxidative enzymes. So these enzymes break down fatty acids, alcohol, toxins via oxidation reactions. So through oxidation reactions, unlike lysosomes that have hydrolytic enzymes that hydrolyzes, right? These ones oxidizes. So they detoxify the cell by breaking down alcohol and other harmful substances, so toxins, poisons. That's what peroxisomes do. But they then produce hydrogen peroxide, and perhaps that's how the name came about, right? Because of the byproduct that occurs as a result of that reaction. But this peroxide that they produce, peroxide is H2O2, hydrogen peroxide. can also be broken down by catalysts to prevent damage. So somebody asked a question about aging. Catalyst is an antioxidant, right? That can help to, so in fact, sometimes we monitor the level of catalyst in animal models, because if your catalyst level is low, that means that it cannot scavenge these free radicals, or this peroxide. It cannot remove it, and you want it to be removed. And the more peroxide you have, the more free radicals you have. The more free radicals you have, the more DNA damage you can have, right? So it could lead to DNA damage. It can repair is not possible. It leads to things like from even oxides, peroxides. They can even destroy the cytoskeleton, spindle fibers, and all those kind of things. So which we don't want, right? We don't want all of that. So that's why you have catalysts that helps to break down this peroxide. And that's why they say you should eat fruits that are rich in antioxidants that can kind of lead to a form of balance between the free radicals that are being produced and the normal antioxidants that you're supposed to have. Okay. All right, so let's just move on. So we have vacuoles. We have different types of vacuoles or vacuoles. I don't know how you pronounce it. I pronounce vacuoles, right? Some people say vacuoles. I don't know, but vacuoles. You have central vacuoles, you have food vacuoles, you have contractile vacuoles. You have central vacuoles in plant cells and fungal cells, including yeast. So these plants mostly have central vacuoles. And so if you look at this figure here, you see the vacuole very large. It's called a central vacuole. And they are basically large water-filled spaces. So you have cell, and they occupy between 30 to 90%. So it varies, right? The volume central vacuoles occupy ranges from 30 to 40 to 90%. And vacuoles have a membrane, but their own membrane is called a tonoplast, and it is a single membrane. So remember I told you that I told you earlier on that membranes have bilayer, right? And that sometimes you could have like two membranes, but the membrane surrounding vacuole is single, only one membrane, and it's called a tonoplast. And so there are different functions of vacuoles. They help to store proteins, to store ions, pigments, acid, salt, waste. So basically, they have storage function. Number two function of vacuoles is that they help to deposit metabolic byproducts. Vacuoles also help with defense. So they store compounds that can repel herbivores, right? Herbivores are plant eaters, right? From the word herbivores, carnivores, herbivores, omnivores, and all of those things. So vacuoles also have these compounds. Remember that we said they have all these compounds. So some of these compounds can deter or repel herbivores from them. And then vacuoles also help to maintain two-gut pressure. And two-gut pressure is very, very important because it's the osmotic pressure that pushes outward on the cell, and it keeps the plant from wilting. So you want rigidity, right? You don't want loss of water. You don't want wilting to occur. So it is the vacuole that helps to maintain this two-gut pressure. And like I said, it also has its own membrane, a selective barrier. Remember when we talked about the functions of membrane, we said it acts as a permeability barrier, right? Permeability and boundary barrier. So these vacuoles have the tonoplast, which controls what goes in and leaves the vacuole. So remember I told you that that's one of the functions of membrane. They serve as a gate that controls what enters and leaves the vacuole. So, but the membrane of vacuoles is a single membrane. It is a single membrane called tonoplast. And this single membrane only has a single lipid bilayer, not two. Remember that we said that other membranes have phospholipid bilayer. So this one has a single lipid bilayer that controls what goes in and out of the vacuole. So on this, in the figure on the right, you have the cytosol, you have the vacuole, you have chloroplast here, and then you have the cell wall. The single membrane is called the tonoplast here. And of course, you can see the size of this the scale bar. So I told you there are other types of vacuoles. So you find central vacuoles in plants and fungi, but then you find food vacuoles in protists, like amoeba, paramecium. Those of you who have drawn or who can draw and label paramecium and amoeba, you recall that you must have drawn things like contractile vacuole, food vacuole, and food vacuoles are formed by phagocytosis, which has to do with engulfing, eating, right? Phagocytosis. So they engulf. We use the word engulf particles or engulf different things. So even phagocytosis is also present in apoptosis, right? So you have dead cells, right, or unwanted cells. They get engulfed by a bacteria, right? They get phagocytosed. That's what we use. So they eat up all those things. So you also have food vacuoles that is formed by phagocytosis, and they store ingested nutrients. Then you have contractile vacuoles that helps to balance the of course, it's involved in cellular homeostasis, so you want a balance. You don't want too much water. So it pumps out excess water to prevent the cell from bursting, which has to do with like osmoregulation, right? So like I said, these vacuoles are present in protists. Remember, we're looking at cellular organelles, right? So, but in plants, you would find central vacuoles. Please note in amoeba, paramecium, and other protists, you would find these other types of vacuoles. I'm not going to go over this table, but like I said earlier on, it just summarizes. I saw it recently, and it summarizes or it lists several structures in plant and animal cells and their function. So it's like a key point. It's like a key point, and I think the only one we've even talked about cilia and flagella. As regarding motility, and I told you that they have. So you for example, sperm cell, you find flagella. In ciliate, you will find cilia. So you look at the cytoskeleton, but when you talk about motility, you know that plant cells are immobile, right? They are immobile. So mostly animal cells are mobile. All right, so you can just look at these and look at their functions, the function of each organelle. So we want to look at plastids. Plastids are also found in plant cells. They are membrane-bound organelles found in plant cells. And they contain pigments or storage materials. So when you talk about pigments, or storage materials, and you would find them in plastids. And there are different types of plastids based on their functions, based on the pigments that they contain. So for example, you have chloroplast, which is very popular and common. You have chromoplasts, and then you have leucoplast. Please note the three major types. There are other types, but these are the three main types: chloroplast, chromoplast, and leucoplast. They are plastids. Now, if we look at chloroplast, also originated from the term chloroplast has Greek origin, and is from two words, chloros and plastos. Chloros, green. So remember we talked about pigments, right? Different pigments. So you have green, some of them could be yellow and colorless. So we're looking at chloroplast, green, green pigment, And of course, you know that that's what is very, very important for photosynthesis. Remember, I told you earlier on that we're going to scratch the surface of photosynthesis. You know the definition of photosynthesis from secondary school, process by which green plants, manufacture their own food. So CO2 plus what? Eh? Okay, let me allow you on mute so that it doesn't, because I've been talking a lot. Where are you guys, participants? All right. Okay, so if you want to talk, you can. Who wants to define photosynthesis for us? You can raise up your hand. 2 marks for the person, the first person to raise up their hand and talk. 2 marks. Yes. Samuel Obi.
Morning, sir.
Yes.
Photosynthesis is by which green plants convert sunlight to food materials.
Okay. All right. Thank you. Anybody else?
So photosynthesis can be defined as a process by which plants uses sunlight and water to manufacture their food, which leads to the release of oxygen and glucose.
Okay, good. Who else? Who was that? Who was the last person? What's your name?
The process by which green plants use sunlight.
The last person. Who was the second person?
Awal.
No, the second person. Who was the second speaker?
Odeyinka Awal, sir.
Odeyinka Awal. Okay.
Yes, sir. Awal.
All right. So one more person, a female. Yeah, who is this female that was speaking? Yes, tell us your name. Yes, a female was speaking. Who is the female?
Hi, sir.
Go on. What's your name? Tell us your name.
Shotunde
Oluwaseun. Yes.
Yes.
Go on.
Okay, photosynthesis is the process by which green plants use sunlight, carbon dioxide, and water to make their own food. That's glucose, releasing oxygen as a byproduct.
All right. Thank you. Thank you so much, everybody. So you can mute your mic. So I already know that you guys should know this because it's a major question at WAEC level, something that you guys, some of you have crammed, some of you, in fact, I think when we were in secondary school, we even had a song for photosynthesis. So, but the last person that defined actually gave the complete definition. So you see CO2, water, and sunlight, right? And then leading to the production of glucose and oxygen as a byproduct. So that's why when you have plants, the byproduct is oxygen, and that's what we breathe in. What we release out, which is CO2, is what they need, right? So anyway, so chloroplast is the most widely occurring plastid in plants, and it is responsible for photosynthesis. And like I said, like mitochondrion, it's also a double membrane bound organelle. It has different shapes. There are different shapes, just the same way you have different shapes of mitochondria. Who can tell us various shapes of mitochondria that we mentioned? Or one or two? You guys have not been reading. Okay.
Sausage shape.
Sausage, yeah. Which other shapes?
Rod shaped.
Rod, yes. Which other ones? Oval, yes. Rod. I can see the answer in the chat box.
Spherical shape.
What did you say?
Elongated.
Filamentous shape.
Thread-like shape.
Rod-like shape.
Vesicular.
Okay, all right, no problem. Just go back to your notes. All of you have mentioned a few, but then you see the different shapes of chloroplast: disc-shaped, cup-shaped, spherical, oval, discoid, ribbon-like. And if you look at the figure on the right, you can see some of the shapes: girdle-shaped, ribbon-shaped, oval-shaped, etc., etc. Now, please note, chloroplasts are mainly found in mesophyll cells of leaves. So you find them mainly in mesophyll cells of leaves, and they contain chlorophyll and accessory pigments, so carotenoids, but major pigment is chlorophyll, right, which is responsible for that green color that you see. So you have them in mesophyll cells of leaves. And like we said, they trap light energy for photosynthesis. And this has to do with the structure of, I think this should have moved to the next slide. So I'll just show you the internal structure of chloroplast. So this is the structure of the chloroplast. So you have the outer membrane, you have the inner membrane, you have the intermembrane space, similar to the mitochondria. So in fact, it is believed that the chloroplast and mitochondria evolved from an ancestral bacteria. So there is the endosymbiotic theory, right, that explains the evolution of chloroplast and mitochondria. And even chloroplast, some people even say that mitochondria evolved from chloroplast, because you have chloroplast in algae, right, cyanobacteria, and different algae, blue-green algae, right? So you have the intermembrane space, but you then have, instead of the matrix of the mitochondria, we have the stroma. So the matrix, these are questions. A question could be, the matrix of the mitochondria is referred to as dash in a chloroplast. A, thylakoid, B, stroma, C, outer membrane, D, starch granules. What would the answer be?
Stroma.
Stroma, good. So the matrix of the mitochondria is synonymous with the stroma of the chloroplast. So you have stroma, and in the stroma, you have chloroplast DNA. The same way the mitochondria also has its own mitochondrial DNA. Chloroplast has that's cpDNA, chloroplast DNA. And then it also has its own ribosomes. It's not shown here, but yes, it's here. This figure doesn't show it. Sometimes some figures are limited, but it has its own ribosomes, and it is also 70S. Remember, when we talked about bacterial ribosomes, I told you 30 and 50S, but then it adds up to 70, not 80. So the chloroplast also has 70S ribosomes present, and it has different enzymes. It also has a starch granule present. And what else? So the stroma lamellae. So you have thylakoids. So thylakoids are flattened disc-like chlorophyll-containing membranes. Each of these, so one of these is called thylakoid. As they stack on each other, it forms what we call a grana. Forms what we call grana, singular granum, right? So it forms grana. So this is grana. And then one of these flattened disc-like structure containing chlorophyll is called thylakoid. And so when you talk about photosynthesis, I'm not going to delve too much into photosynthesis because I want to give you as an assignment, I want you to read up, although you will still do this in botany. So there is what we call the light reaction and the dark reaction. I don't want to give expo too much about what happens in the grana and in the stroma, but linking the grana together, what links the two grana two grana plural together is called the stromal lamellae, which is this line here, is what links two grana together. It's called the stromal lamellae. So I'm going to come to the slide that has your assignment in a moment. But this, again, basically is the structure of the chloroplast. And your questions could also be diagrammatic. So from your test, you would see most of the things I have said. I've said a lot of things that you might not find on the slide. And I've tried to give you like likely questions, even during the lecture. So those of you who have listened will be able to answer them. Those of you who have not, we have put up the link to the recordings. Please watch them, particularly the YouTube recording. And I observe that many of you are not following me. I mean, you've not subscribed to my YouTube channel. I don't know why. So you need to subscribe to my YouTube and watch the YouTube video, because like I said, the videos have been uploaded there. All right. And you'll be able to get some of these questions that I said. I'm also going to go back and watch the recording. Those things I stressed, those things I emphasized are likely questions. So you have other types of plastids, not only chloroplast, but chloroplast is the major one. In fact, the diameter of chloroplast, I don't know if I mentioned it, is around 5 to 10 micrometer, but maybe this figure shows it. Is there any figure that shows? Okay, this one doesn't show, but it's usually around 5 to 10 micrometers in length for the for chromosomes. But you have chromoplast, and like I said, chromoplast, unlike chloroplast, which is green, they provide plant parts with yellow, orange, and red colors. Yellow, orange, and red colors, and you find chromoplast more in flowers. So you see all these yellow flowers that are beautiful, orange flowers like Pride of Barbados. You know, orange flowers, yellow flowers, leaves that are aging. You know, sometimes when you have plants, I have some plants in my office. Okay. I wanted to show you one old plant. Yes, I found this one. So if you look at this, you can see this leaf, you know, so it is now old. So you can see that definitely the chlorophyll level is low. So what you have will be chromoplast, right? Unlike these other ones that are green. So I'm showing you, these ones are green, so you see that it has rich chlorophyll content, right? I mean, I'm not a botanist, but we have plants in the office, so I just thought to show you since my video is on today anyway. But you would find chromoplast in fruits, you know, in carotenoid pigments, so carrots, even in carrots, all those kind of things. So they have carotene. They are chromoplasts. That's the type of plastid in those fruits. So carrot, pawpaw, and all those kind of things. Then you have leucoplasts. Leucoplasts are colorless. They are colorless plastids, and they are primarily involved in the storage of nutrients. So you have examples include amyloplast. Remember I told you that you have different types of plastids. So you also have amyloplast, which stores starch. You have aleuronoplast, which are proteinoplasts. They store proteins. You have elioplast, which stores and fats. All of these are colorless. So they are broadly categorized as leucoplast. But when you look at leucoplast, different types, you have amyloplast. This one stores starch. So if you look at potatoes, you know, they are not really colored, as it were. So you say they are colorless plastids. And then you have proteinoplast. This one store proteins. Then you have elioplast. They store oil and fats. All right, so that's the much I wanted to say. This is the assignment for this class, and that brings me to my last slide and the end of the lecture. I'm going to take a few questions from you guys. So the assignment is to write a short essay on the role of chloroplast in photosynthesis. I want you to include what happens in the light reaction and the dark reaction briefly, and if you the structures of the chromosome. So I've shown you the internal structure of the chloroplast, all of this, the thylakoids, the grana, the stroma. So when you're writing your essay, I want you to focus on the structures of the chloroplast that are involved in photosynthesis, and then you have some electronic, so it will be easy for me to count, right? If it is more than 500 words, please include your word count. So at the top of your essay, write your word count: 480, 492, 499, include your word count at the top. If it is more than 500 words, it will not be marked. Automatically, you will get 0, irrespective of what you have written. You can include 1 or 2 figures if you think it will enhance your work, but it's not compulsory for you to include figures. And then you're saving it as a PDF file. You're going to submit class by class. So your class rep will create a Google Drive folder and then put the link. So I want the CBG class rep to collate a list of all these links and send to my email. So there will be one document. So only one person is writing me an email. Please listen to this instruction very well. Only one person, which is CBG class rep. It will collate all the links, right, in one Microsoft Word document, the link to all the departments. So you just write chemistry department or whichever department in this MBBS, you put the link to their Google Drive folder. Marine, you put the link like that. And then in that Microsoft Word document, you send it to my email on the 30th of January by 12 noon. This means that all class reps should have sent that link. Is a link you can create today, right? Should have sent that link to the class reps so that on 30th, but any link you send, please, after 30th, no other person should be able to add to your folder. So you go and lock the folder and prevent people from adding more to it. And even if you don't do that, once I download all the responses there, if your own is not there and you tell me, oh, my class rep forgot to pull my own, or you removed my own, I won't listen to it. Once I download all the responses I need, that's it. So class reps, please make a list of those who submitted before 12 noon, who uploaded to Google Drive folder before 12 noon. Send it to your class group so that nobody will say, oh, I submitted, it was not there. I'm not going to listen to things like that. So you have 2 weeks to do this assignment. It is inside your document that you have to write your name, your matric number, and the name of the document itself should just be your matric number. Don't name it with any other thing other than, so that by the time I check your folder, it will arrange it from matric number 1 to matric number 100 if you're 100 in your class. All right, that's it. So let's take questions, 1 or 2 questions in the next, how many minutes do we have? 4 minutes more. Do you have questions? I want you to, instead of typing it today, maybe you can just quickly unmute to ask your questions. Please subscribe to my YouTube channel. Those who subscribe, so this assignment is over 20. Those who subscribe to YouTube channel would automatically get 5 marks. Those who watch the YouTube video will get another 3 marks. So that's 8 marks. So the assignment itself is 12 marks. So I just also wanted to say that. So you can already get 8 marks by watching. So the views will count. When you subscribe to the YouTube channel and stay subscribed, you get 5 marks. You watch you get
your
3
YouTube
marks.
channel.
So you watch each video. So you know there are 3 lectures. Lecture 1, lecture 2 is the same link. I sent the YouTube link to your class group. If you watch lecture 1, lecture 2, 1 mark, lecture 3, 1 mark. And you must watch it from the beginning to the end. So you have 1 hour. So that's 3 hours. I would see it. You know that this thing.
Serious about this.
I'm serious about it. Ask your colleagues. When I say things, so you already have 3 marks for doing this. For watching, your watch hours will give you 3 marks.
How do you know, like the person that actually watches it?
Your subscription, don't worry, I will know. I've told you I will know. So, and then you get let's take questions.
Yes, sir. What if you are watching from Zoom only? Do we have to still go to the YouTube channel?
If you are watching from what?
Zoom.
No, not Zoom. The YouTube is different. I'm not talking about Zoom now. The YouTube watch hour is what I am interested in. So if you watch lecture 1, you get 1 mark. If you watch lecture 2, you get 1 mark. If you watch lecture 3, you get 1 mark.
Excuse me, sir.
Yes. Sir, I want to take questions related to the lecture before. How will I what?
Hello, sir. Hello, sir.
Before you ask your question, please raise up your hand. Let me call you, please. Use the raise hand button so that we will be organized. I would have Ajayi, after Ajayi, Smith, and then Opeyemi. Ajayi, unmute quickly, please.
Good morning, sir.
Yes.
Good morning, sir. So I wanted to ask, is there any importance of the intermembrane space in the chloroplast?
Well, there should be. The electron transport chain allows for, in the mitochondria, for example, protons are pumped into the intermembrane space, but in the chloroplast, the electron transport chain doesn't occur in the inner membrane of the chloroplast. So, but I believe that it occurs, I think it occurs in the thylakoid, I'm not sure. But definitely, protons would also be pumped. The intermembrane space too would also have its own functions. But I am not sure because the inner membrane of the chloroplast is not where the electron transport chain occurs. So I am not sure exactly what the intermembrane space might be doing in the chloroplast, but every part of an organelle has its own function. But definitely, I think that the protons that will still be pumped from the electron transport chain, you know, so in plants, you have things like photosystem 1, photosystem 2, all those things. I really don't know exactly now what the intermembrane is, but definitely some of these electrons and protons and all might be passing through the intermembrane space as well.
Okay.
Smith.
Thank you, sir. Sir, I have another question, sir. Last one.
We don't have time. It's 9.
Thank you. Okay, so I wanted to ask that is the peroxisome part of the endomembrane system? Is it still considered not part?
Peroxisomes.
Yes, peroxisomes. Sorry, sir. Yeah.
Lysosome. I told you there are 7 of them. Where are they? Is it there? Lysosome. I know lysosome is there. Vesicles, vacuoles. No, it's not there. It's not there. Okay. So you have nuclear envelope, ER, Golgi, lysosome, vesicles, vacuoles, and plasma membrane. So it's not. All right. Next.
Sir.
Good morning, sir.
Yes.
So please, I want to ask if macromolecule is the same thing as polymers. And sir, secondly, some of your YouTube videos are not playing.
They are not playing, okay? Are you sure it's not from your head? So you can't say macromolecules. I don't know what you mean by polymers, because when you say polymers, you're talking about 2 monomers, and then but then macromolecules basically are DNA, RNA, proteins. That's what we refer to as macromolecules. So even when I showed you the level of organization earlier, you can have atoms, you have molecules, then you have macromolecules. Where is it? Level of organization. Yes, this one. You can see you have macromolecules. So you talk about DNA, proteins, carbohydrate, but then these are made up of monomers. So it depends. So when you have different monomers, then you say polymers. But when you say a polymer, everything that is a polymer might not necessarily be a macromolecule. Do you understand? So, but macromolecules definitely are made up of smaller units called monomers. So, but it depends on what you're looking at, as you know. But proteins, carbohydrate, lipids, DNA, RNA, those are macromolecules. So, but you know, in chemistry, you have other things that are polymers and all, but I don't know. So I cannot say exactly if all polymers are macromolecules.
Good morning, sir.
You understand? But what I do know is that macromolecules fits.
Good morning, sir.
Yes, good morning.
Sir, I want to ask the lysosomes. How do they destroy the unwanted cells? Like, do they engulf them, or like what's the process, sir?
So they can fuse with you know that I told you that vesicles from Golgi, the trans-Golgi network, they pinch off and they are supposed to be transported to the plasma membrane on their way, assuming that it is something that the cell doesn't want. It will go and fuse with the lysosome, and that's why the endomembrane system is very important. So when it fuses with the lysosome, I want to see here. So if you look at this, so you see, let me minimize this. So you see this vesicle. Can you see the vesicle here, the Golgi, from the Golgi, fusing with the lysosome? Can you see it? So it will enter, and then when it enters, the hydrolytic enzymes in it will digest it. So that's what happens. So instead of it going to the plasma membrane, because it has undigested materials, it has things that it wants to remove, old organelles, different things, it just goes to the lysosome, and that's why it is digested. Does that answer your question?
Yes,
Yeah. Good morning, sir.
Yes, sir.
Yes, user always talking now. Faith, please, if you've asked a question, you can lower your hand. Faith.
Sir, it's me that is user.
Okay, who is user? What's your name?
My name is Marion. Marion.
Okay.
Sir, as for writing the assignment, can we use diagrams?
Yes, I said you can include diagrams.
Okay.
Include diagrams, but you are not writing or you are typing, and you must use Arial fonts. You are typing, you are not writing with biro. Nobody should. Once you write with Foolscap sheet and snap it 0, it will not be marked. Please tell your colleagues, once you write with biro, it must be typed. Arial, not just even using any font. You must use Arial fonts, size 11. So the instruction is there. All right. I will just take one more question. You can use the chat box.
Okay,
the last.
Yes.
Yes.
This is a Kwemi class here for pharmacy. I want to remind you that you said you are going to drop the PDF, the slide you are using. You've not dropped that.
I wanted to just finish my lecture before sending it. I'll send it today. I'll send it.
Sir. Thank you, sir.
I wanted to add the assignment, everything, so that by the time I'm not sending slide 1. Some people will be reading incomplete. Do you understand? So, but now I have the full slide. I'm just going to send it now, just this morning. All right, Abdul Basit.
Okay,
the
thank
last
you very
question
much,
for
sir.
today. Yeah. Who else? Okay, nobody. All right, thank you so much. Next week you'll be writing your test, so please study. All right, bye.
Sir, next week on, please, sir.
The date will be communicated to you. Let me check. I know the date has been fixed. I could check for you if you want me to check.
Sir, please excuse me. Can I ask a question, sir?
Yes.
Sir, is it like only biology test or like general test?
Well, I know for biology. I don't know about other departments, but I'm trying to check the dates.
Please, is it a physical test, sir?
The date is January 19th.
January
19th. What's today's date? So that's next week, Monday. Well, it might have been changed, but I don't know. Has it been announced to you?
No, sir.
No, but I mean, tentatively, it's supposed to be January 19th. So please plan.
Sir, is it a physical test?
I'm not sure. The results will be communicated to you. So. All right. That will be all. Have a good day.
I want you to see your children.